Okay, let's now turn our attention to cognitive neuropsychology. This is the study of patients with brain lesions, so some kind of structural damage to the brain, either caused through a stroke, so for example where you have temporary blockage of oxygen to a brain which kills off the neurons in a specific region, or it can involve some kind of traumatic brain injury. And studying people with damage to a particular region can be useful for illuminating the potential function of this region. So for example, we're going to discuss later in this course the case of patients who are unable to recognize faces. Despite their visual areas being intact, they typically have damage to a non-visual brain area, this is known as the fusiform face area, that appears to be specialized for processing faces. Now, cognitive neuropsychologists make some important assumptions. First is the idea of modularity. So the doctrine of modularity proposes that the brain comprises numerous modules or processes that are operating relatively independent of each other. So for example, aspects of visual stimuli, for example the color, the form, the motion, are processed in distinct neural areas. And you can have regions of the brain that are specialized for processing one particular class of stimuli, such as face recognition. Second is the idea of generalizability. So researchers assume that the organization of these regions of the brain is comparable across people. So in other words, although everyone's going to have a slightly different size and shape of their brain, the organization of the brain and how these regions are located relative to each other is going to be pretty similar across different people. Third, there's the idea of subtraction. So this is the idea that the brain damage can impair the functioning of specific modules, but it can't lead to the development of new ones. So one final assumption of cognitive neuropsychology is the idea of dissociations, particularly single and double dissociations. So a single dissociation is evidence that damage to a brain region leads to impaired performance on one task, but intact performance on another, ostensibly related task. So for example, a classic single dissociation is the finding that some amnesic patients might perform very poorly on tasks that involve long-term memory. So for example, trying to remember what they did yesterday, when they last had a meal, whereas they might do pretty well and function absolutely intact on short-term memory tasks. So if you ask them to remember a telephone number and then test them 30 seconds later, they can do this. Now this single dissociation gives you a suggestion then that there might be different modules that are responsible for long-term memory and short-term memory. In other words, that they are distinct processes. But, and this is a really important point, it doesn't necessarily demonstrate, so it doesn't prove that long-term memory and short-term memory involve separate modules. See if you can think why not, what other explanations might there be? Well, one reason is because long-term memory may simply reflect a more difficult form of memory. So if you think about it this way, it could be the case that memory is one single unitary process. And short-term memory, that is the bit that's intact in these patients, is basically reflecting an easy form of the memory task. Whereas the damage that they experience maybe just impairs their ability to engage in the kind of more complex or more difficult form of memory, which is this kind of long-term memory process. So again, following on from that, can you think of any ways or any idea that you might have to show that short and long-term memory involve different systems? One way is to find a double dissociation. And this is where you find evidence that some patients would have intact performance on one task, but impaired performance on another, whereas another individual or group of individuals would exhibit the opposite pattern. So for example, a patient with a lesion in brain region A performs well on task A, poorly on task B. A patient with a lesion in brain region B performs well on task B, but poorly on task A. If we return to the example of where there are different systems for short and long-term memory, we do see pretty clear evidence for a double dissociation there. So for example, you have patients with a form of amnesia called Korsakoff's amnesia, and they will show long-term memory damage with intact short-term memory. So that case that I just presented to you before. However, the crucial thing here, and what shows the double dissociation, is that you do find other patients that have something known as anterograde amnesia, whereby their long-term memory seems to be relatively intact. So they can do what might have been this kind of more difficult, complex form of memory, but their short-term memory, so their ability to retain things over the short term, is severely impaired or damaged. So from this, you can make a pretty strong inference when you get this double dissociation, that there are separate short-term and long-term memory systems. Now, before the era of kind of non-invasive neuroscience methods such as fMRI, we relied on famous case studies of patients to reveal the specialization of specific regions. So, for example, Phineas Gage was one of the very earliest examples to show that damage to the frontal regions resulted in changes in personality and emotions. You have Paul Broca, showed that patients with lesions to the left temporal lobe had impaired language abilities. Henry Molaison, or patient HM, was somebody who had his medial temporal lobes removed due to severe epilepsy, and he went on to develop severe memory issues, revealing the kind of vital role that these regions played in memory. And also in memory, there's a patient called Clive Waring who had hippocampal damage, and then went on to also exhibit some severe memory impairments, and we're going to meet those later in the course, but they're important case studies that illustrate the fact that damage to a particular region implicates their role in specific functions. Now, there are some important limitations to cognitive neuropsychology. First is that it would be easy to interpret the findings from patients if the brain damage was always limited to a single module. However, you just don't get that in real life. If a patient has brain damage through stroke or traumatic brain injury, normally it's across a distributed region. There's no kind of clean, clear cut, we're just going to knock out this one region and leave everything else unimpaired. So generally it's difficult to work out exactly where the regions are impaired or how far this spreads. And there's also no consistency across different patients that you might have. So, in other words, we don't get neat lesions. Now, in an ideal world, if you were thinking of being a neuropsychologist and you wanted the perfect lesion, then the sorts of properties that might have is that it would be small. It would affect only a single module. Again, in an ideal world, you might want this just to occur briefly. So if you could cause a lesion and then turn the lesion off, that would be great. And it would also be ideal if you could go in and select what particular region you want this lesion to affect. Now, we can't just go around creating these lesions by whacking someone on the head, but researchers can do something kind of close to this. So, one of the techniques that we can use to create a sort of artificial temporary lesion is using something known as transcranial magnetic stimulation, or TMS. So here, a coil is placed close to the head and precisely aligned to a specific location. Then a strong magnetic pulse is applied briefly to a small area of the brain. And this effectively creates an artificial lesion in this area of the brain by inhibiting activation of the neurons. And we can use this then to determine whether that brain region is necessary for a specific function. So for example, if you go and place that coil over a region of the motor cortex, and then you ask someone to do a simple action such as touching their nose, as soon as you apply the magnetic pulse, people's ability to move their hand will become significantly impaired and they can't control their arm. Now this approach also, it does kind of sound great, but there are some limitations when we're taking this. One reason is that a lot of times with TMS, we're actually kind of unclear how much cortical activity is affected. So you might apply this magnetic pulse on one specific region, but you then get spreading of this activation. So it's sometimes hard to understand exactly where we're activating. It can also increase brain activity. So some of the effects are not entirely understood.